In complement fixation test, we will learn what are complement factors, briefly take an overview about their functions, especially with respect to immunology, and then we will understand the principle, the performance of the test with the help of a video. So let's learn complement fixation test. Let us take a brief overview of the terms involved in it. So the test was first used in 1909 by Wasserman for the diagnosis of syphilis, and since then it has been extensively used in serology. Complement basically means a protein and it is found in all human beings in serum. So when I say complement, it is not just one protein, but it's a system of number of proteins. Say 9 different proteins are involved into it. So we denote them as C1 to C9. And what these proteins do is they carry out a chain of reaction one after other so that the final product or final outcome of those reactions is in the formation of a membrane attack complex which actually attacks the cell membrane of the invading pathogen by lysing the pathogen. And therefore, it is extremely important immunologically. When we study complement system in detail, we realize that there are a number of pathways by which these complements get activated and process the reactions. However, for today's lecture, we will not go into the details of the pathway, although I will give you a pictorial overview of those pathways in the next slide. Today, we will see that these proteins, how they are implemented, how they are utilized, how their properties are applied for diagnosis of infections. So the most important property of these proteins are they are very much heat sensitive, they are heat labile, and they are easily destroyed at 56 degrees Celsius in 20 to 30 minutes. More important property is these complement factors bind to antigen antibody complexes. So that is also a very important property because of which we can utilize these complements in the serological testing. And last but not the least, if we change the pathogen with RBCs, ultimately it is going to lyse the RBC. So this helps in visual detection and therefore it helps in forming the indicator system which we use in complement fixation test. So I hope somewhat you have got an idea what are complement proteins, how many complement proteins are there, what exactly they do. This is the picture that I was talking about, that we have C1 to C9 different complement proteins which together come together and form a membrane attack complex. You can see it here. And this is a microbial cell into which you have got a membrane attack complex embedded through which the cellular constituents will be coming out and eventually it will lyse the microbe. So as you can see it on the left hand side, there are different pathways by which complement gets activated. It could be a classical pathway, it could be alternative pathway, or it could also be a lectin pathway. However, irrespective of pathway, the final outcome is the same. Moreover, you can also see that there are certain complement factors like C3, C5 which are getting split into two parts, right? So those parts which are not actually following the path in the reaction are called as anaphylatoxins, and they also carry out important function like inflammation. So these are the basic information that I could give you about complement. Since the discussion is about test, let us move ahead for actual learning about the principle of complement fixation test. So as we just saw, complement takes part in immunological reactions and it requires combination of antigen antibody. This property is used in this test in order to fix the complement. Fix the complement meaning in order to attach the complement to something. Okay. We will understand it more in detail as we proceed with understanding of the systems used in test. So we use a system called hemolytic system. Now why it is called as hemolytic system? Because it comprises or it consists of RBCs which are going to lyse, and this is also called as indicator system because whether the RBCs are lysed or they are not lysed will indicate the results, will help us interpreting the results. So it is also called as indicator system. Now what this system comprises? It comprises of RBCs, but whose? It is ship RBCs. And then we also need to have specific antibodies targeted against these RBCs. So how do we get them? We get them by inserting these ship RBCs into rabbit, and rabbit giving us the antibodies against these RBCs which are specifically called as amboceptors. So the hemolytic system in together comprises of two components, ship RBCs and amboceptor, which are antibodies against ship RBCs. If the complement is fixed, then there is no lysis of ship RBCs, which indicates a positive test, and if complement is freely available, then there will be hemolysis, which will indicate a negative test. We will understand what does these two statements mean when we actually learn the test. So the test requires in total 5 reagents, and the test is carried out in 2 important steps. Let us for the time being not understand the steps and first understand what are the 5 reagents that we need to have. The 5 reagents that we are going to take are also made in 2 different systems. One system we already learned, that is hemolytic system, which already had 2 components. Now we are going to learn about another system that we use, which is also called as a test system, which comprises of 3 components. Component number 1 is the antigen, which we all have to obtain commercially, against which we have to test the patient. So the antigen could be soluble or particulate. It could be a bacterial surface antigen or a viral antigen. It could be anything. The second component is the human serum. Human means patient here. So whoever is under test, that patient serum is required. Now here it is noted as antibody, but we are not sure whether the serum sample has the antibody or not, and therefore we should always say that patient serum. Depending upon the presence or absence of antibody, we would get the results. So let us understand how the results vary. And third and most important part of the test is here in the test system, that we add complements. Complements from the guinea pigs. Please understand that we have to get a freshly obtained complement always. That's a better for any test, because as we said that the complement factors are very much heat sensitive, and so we need to preserve them at minus 30 degrees Celsius. But it's always better to use a fresh complement system than the stored one. And if you're using a stored one or a fresh one, you are supposed to test and standardize their activity before using in the test. And after the test system, we have to add indicator system, which is nothing but a hemolytic system, which we just now talked about. So I hope the 5 different components used in complement fixation test are clear. Let us see what happens in positive test. As I said, we are going to perform 2 different steps. In step 1, we mix the commercially obtained antigen with patient's serum. Suppose that patient's serum has antibodies against this particular antigen, and then we add a complement. We provide sufficient incubation time and temperature regime. The complement will get attached to the antibodies. which are specific against antigen. So first antigen antibody complex will be formed and complement will bind to the antibody. So we say attachment of complement, we note it like complement gets fixed. And why we call it as a fixed? Because then it is unavailable for step 2 in which we add hemolytic system. And I hope you remember hemolytic system comprises of RBCs and amboceptors. So we are providing ready-made antigen antibody complexes, but complement is not available because antigen antibody complex is made already in step 1. So complement got fixed and it is not available to bind to AMBO receptor and RBC complex. So even if you give the same condition, complement is not available, there is no hemolysis and the test means positive. On the other hand, in negative test, you can see that antibodies are absent in the serum sample, complement is not getting fixed, so complement is available for hemolytic system to get fixed onto, and then you see a hemolysis. After 1 hour, the test, if you see hemolysis, the test is called as negative. Let us understand with the help of a video. So most important criteria that we have to follow before we actually start with the complement fixation test is we have to heat the serum sample at 56 degrees Celsius to get rid of any of the complement factors that we have got in the serum sample. We don't want them to interfere with our results and therefore we need to heat. So here on left hand side you can see a patient serum with antibodies against a specific antigen, while this is another patient serum which does not contain antibodies. Let us see what happens next.
The complement fixation test is used to test for the presence of specific antibodies in a patient's serum. Patients differ in the amount of complement in their serum. To standardize the complement fixation test, the first step is to heat the serum to destroy the patient's complement. A measured amount of complement and antigen are then added to the serum. In patient A's serum, the complement is fixed by reaction with the antigen antibody complexes. In patient B's serum, there are no antibodies to complex with antigen and the complement remains free. To determine whether complement has been fixed, sheep red blood cells and antibodies against sheep red blood cells are added. In patient A's serum, the complement is already attached to the antigen antibody complexes in the serum and cannot induce sheep red blood cell lysis. Consequently, the red blood cells settle to the bottom of the tube. In patient B's serum, the complement is free to react with sheep red blood cells antibody complexes, resulting in lysis of the sheep red blood cells. The serum turns a pink color as the red blood cells lyse.
So I hope the animation has helped you sufficiently to understand the complement fixation test. This is a YouTube video freely available for learning purpose. Important is the interpretation, and this is how you get to see in laboratory. When RBCs settle in the bottom, you actually get a red button like appearance at the bottom of each well. And so we say there is no hemolysis. No hemolysis interpreted as positive test. Hemolysis, as you can see in 4A, 4B samples, it is actually interpreted as negative test. I hope you all have understood complement fixation test, and for any doubts and difficulties, you can comment on the video. Thank you.